Yeah, madam, we can start.
Good day to everyone. It gives me an immense pleasure to extend to you a very warm welcome on behalf of Enviro Club in association with Department of Plant Biology and Plant Biotechnology at Sri Chandra Prabhu Jain College, who has welcomed all of us with regard to the celebration on World Food Safety Day 2023. I welcome our esteemed principal, Dr. N. Sujata ma'am, who motivate us to organize this session. I welcome our Enviro Club coordinator, Dr. V. Kokila ma'am, and the team members. I also extend my welcome to all heads of the departments and faculty members and my dear friends. Now, I would like to invite Ms. S. Abhinaya from Department of Plant Biology and Plant Biotechnology to deliver formal welcome address.
Good afternoon to everyone. On behalf of our college management and Enviro Club, I would like to extend a very warm welcome to our respected Principal Dr. N. Sujata ma'am, who is in absentia. And I welcome our Enviro Club coordinator, Dr. V. Kokila ma'am, and Dr. C.P. Prakash sir, and Dr. B. Bharat sir, assistant professors, Department of Plant Biology and Plant Biotechnology. A special welcome to our chief guest, Dr. J. Vardhana, Assistant Professor, Department of Biotechnology, Vels Institute of Science Technology and Advanced Studies, VISTAS, Chennai, who has accepted our invitation to be the keynote speaker on today's auspicious occasion. I also extend our welcome to the heads of various departments and all the faculties who joined here. It's with great pleasure that I welcome all our students and friends. It is truly privileged to have the opportunity to learn from such experienced and knowledgeable educators. As students, we constantly seek ways to expand our knowledge and improve our understanding of the world. This webinar is a perfect opportunity for us to do just that, as we can hear from experts in various fields and learn about new and exciting research to the students here today. I want to remind you to make use of this opportunity. I won't keep you long, and let's begin the day. I wish all of you a valuable and wonderful time. Thank you.
Thank you, Abhinaya. Now, I would request Dr. V. Kokila ma'am to deliver chief guest introduction to the gathering. I welcome you, ma'am.
Thank you, Bakia. It's my pleasure to introduce our chief guest, Dr. J. Vardhana, Assistant Professor, Department of Biotechnology, Wales Institute of Science, Technology, and Advanced Studies, Chennai. Dr. J. Vardhana is a remarkable individual who has achieved great success in the field. She is a true inspiration for many of us, and we are honored to have you on this day. She is a dedicated professor in her specialization. Coming to educational qualification, she did her undergraduation, post-graduation, and doctoral program in biotechnology. Apart from this, she has obtained DST Inspired Fellowship from 2011 to 2017. She has 6 years of teaching experience in the field of biotechnology. Besides, she is also act as an assistant editor in International Journal of Pharmaceutical Science and Research and editorial board member in International Multidisciplinary Conference in Research and Innovation. In her career, she has attended and presented various papers in national as well as in international conferences and workshops. So far, she has published 15 papers in the reputed journals. I'm very grateful to have you on this day, ma'am. Once again, I welcome you. Thank you.
Thank you, ma'am. Dear all, it is a great opportunity to listen this session. Yes, it will be my honor to invite our chief guest to deliver her thoughts about the theme of the program. And the session is over to you, ma'am.
Thank you so much. Thank you so much for the wonderful introduction, Dr. Kokila. And now I would like to share my screen. Am I audible, all of you?
Yes, ma'am, it's
Yes, ma'am.
audible, ma'am. Ma'am, you can start, ma'am.
Yeah. Is my screen visible?
Yeah, ma'am.
Okay. So the topic for today will be about quality testing techniques in food industry. So we all know that humans, we consume food every day, and we are going to just see few things which we need to concentrate in the food which we are going to eat, and we are also going to see the standards that is being followed in our country to grade the food which we are consuming in our country. So coming on to the introduction, we all know that food is a basic need of human apart from shelter and clothing. So quality control is a process in which we are going to examine the food whether it is good or bad. So it is an essential component in any food business, in any food whatever, even if we are doing a small food business, quality control and quality assurance is an important thing. And food analysis is a process for both fresh as also the processed food, the food which we get in packets, all the biscuits, chips, everything, or other processed foods like frozen peas, frozen meat, everything. All these are considered as the processed foods, and some other packaged drinks. Everything has to be analyzed before it is coming into the market for human consumption. And these analytical techniques gives us whether the food which we are consuming is of good characteristics, which includes their nutritional value, their properties, and how far it has to be consumed, the expiry and everything. All these are the things which will be coming under quality control and food analytical procedures. So why we need to do this food testing? So first and basic thing is it is to protect the consumers and customers from the danger and ensure that they get the weight and quality of the food that they pay for it. For example, if we are going to some shops, the measurement which they are giving will be different from the one which will be asked for. So all these small things will be coming under the quality. So this is the main thing for which we require the quality control. First, it need to be without any toxic free, and it should be safe when we are consuming. Second, it should be of accurate weight and the quality which we are paying for. Second, it is to protect the business. So the main thing is about a life, and the second one is giving a business to someone without cheating them by the suppliers or damage to the equipments and false accusations by middlemen, customers and suppliers. So whatever we are getting from the market, it is not directly coming us to the manufacturer. It is going to the in between intermittent parties, and from them only we are getting it. So in order to avoid all these chaos between all these people, it has to be undergone testing and quality control. And it also it should be ensured that the laws under which the food operating or the food which we are coming, and it has been operated under the laws which is provided in our country. We know that Hallmark, BSI, ISO, all these some of the standards which is being followed in our country to grade the food or food testing and food quality. So what are the quality parameters we should consider when we are going to check any, or when we are going for any food testing? So the first quality parameter is, in order to ensure the right quality of various food products, several parameters has to be evaluated. First is a physiochemical and rheological parameters. Physicochemical in the sense it is about the color, texture, moisture, water content and density, and all these things will be coming under the physicochemical property. And phytochemical parameters, it is the nature of the components which is present. Phyto means plant, and the components which is present, any good nutritional value which is present in the plant, and what are the components. We will say, some food is rich in vitamins, some food is rich in this protein. So how all these conclusions are being taken with the help of these parameter analysis. So we will be checking the physicochemical and rheological parameter. Next is your phytochemical parameter, and the final one is your packaging material. What are the components which is used to package the food material, the material which on from which the food is packed, what are the components it has, whether it is having any heavy metal, and what about the permeability, and what will happen when it is reacted with heat. All these things will be considered, and these are some of the quality parameters which has been checked. Quality, we all know the term quality is the main thing for anything, for any of the product, not only food, for anything which we are doing, it requires a quality. Even when you are taking for studying for examination, it requires some quality, we will say. So quality is the one that is a major criterion for anything which we are doing. So when you are taking food is the main thing which helps us to live and which helps us to survive. So the quality control that has to be evaluated in the food by two methods. First is your sensory method, and the other one is your objective method. Sensory method is base of senses. So senses, subjective analysis is your method of sensing like smell, taste, odor, and all these will be coming under how it is. So it is a basic method for evaluating the food quality. First is your sensory method. So some methods of perception by human senses, we will say sometimes we will smell some food and we will say that the food has been spoiled. Sometimes we will say the smell is very pleasant, this smell is coming from that. So all these are some of the quality checking which takes the sensory methods. So sensory analysis or sensory evaluation, it is accessed by human senses, by sense organs. Example, appearance, what is a color, appearance, how colorful your food is, and how it is been appearing, whether it is hard, soft, soggy, or whatever it is. So that is one first thing, appearance. Second is the flavor, next color, and the final one is the texture. So sensory analysis is also called a subjective or organoleptic test, and there are three subsections for this. First is effective testing, affective testing, and perception. So coming on to effective testing, what are the it exactly deals with the product, and the product when the from which it has been made, or what are the facts about the product. If you take some packages, you can, if it is green color, we can assume or we can say that it is purely made up of vegetarian or veg. If it is coming with red, you can see anything, it is any of the animal product is involved in that one. Sometimes they may use animal fat, and all these, if they are using, they will be marking it with the red. So all these are some of the facts about the products, and that is called as effective testing. Affective testing, subjective facts such as preferences, that is how well the products are likely to be accepted, and it is also called as consumer testing. So for example, affective testing, if you are going to prepare a food product with this component, whether the people who are going to consume that food will accept it in a larger way, or only small group of people will buy that one. So the testing upon considering your consumers is called as affective testing. Perception is nothing but biochemical and physiological aspects of sensation. What are the components which is present? Biochemical means what are the biological components, whether it is rich in vitamins, proteins, fats, or all the, or some of the chemicals or biochemicals which is present. So this is called as perception. So sensory method is dealing about three things. One is effective testing, affective testing, and perception. So objective methods, the second method is your objective method. It is the one which is done other than human senses. So apart from appearance, color, and all those things, these are the method that is done apart from human senses. For example, characteristics like density, maturity, moisture content, oil content, tenderness, all these are considered as the characteristic that has to be analyzed using the objective method. So if you take the objective method, we have 6 different things. One is physical property, mechanical property, optical property, electrical property, magnetic property, and electromagnetic properties. So now we'll be seeing one by one. Physical properties such as shape, weight, specific gravity, bulk, density, volume, porosity, all these are measured with the help of your physical properties. and these are easy to understand and perceive, and it is easy to determine and does not require the sophisticated instrument. Physical property, we are just going to take the product in hand, just we are going to see the appearance of it, and then we are going to see the physical nature of it, and then we are going to determine what kind of product it is. That is called as physical property. Second one is electromagnetic property. Here we will be using the wavelength between 400 to 700 nanometer. It is a kind of spectrophotometer where we will be measuring the wavelength of some product to test the components which is present in it. So depending upon the wavelength or depending upon the peak, we can say that this compound may be present in the food product, or this may not be present in the food product. Next is your optical property. Appearance gives first impression about the quality of the product. The optical properties measuring appearance and texture of a material are defined by the percentage of incident light that is reflected, transmitted, or observed by each wavelength. So here they are going to use the wavelength to determine the property of a food. For example, if it is appearance, you will also come under, it is also coming under the color. Color is an appearance and texture. So the amount of light that is being observed by the food product will determine the property, and that is called as optical property. So defining the color is very important during the quality detection. So to define the color of the food product, we will use a thing called as colorimeter. In the colorimeter, you will be having different wavelengths for different color, and the most commonly used is your red, green, and blue color. Detection of these is having a different wavelength, and with the help of the wavelength or a peak emitted, or with the help of the result we get, we will say that your food is rich in this color, or your food may be having some of the adulterants like coloring, artificial coloring agent would have been added. All these can be detected with the help of this optical property. So acoustic property, acoustic, the word acoustic means sound. Okay, so for this sound, they will be using, for example, if you are going to get coconut from the shop, what we will do, nama generally thirty paapom thengaava. So that is a property, one property that is giving the what that we are going to determine based upon the sound. Not only coconut, we have so many products. For example, apples, eggs, all these will be having some property depending upon the sound which will be measuring. So based on acoustic response generated by the product, and we'll be using the ultrasound waves. So higher power, lower frequency ultrasound excitations, it is used to determine the tissue, fruits tissue property, and it is also used to grade fruits and vegetables. And if you take the limitation of this method is the contact between the acoustic probe and the sample is may be different. So it is not sure that it is having a good contact. Sometimes a probe may be giving you the false positive result or false negative result, or it may give you the wrong result. So affected by surface characters like cracks. So if you have any cracks on top of the food product, the cracks may affect the sound, which will give you the false result. So these are the limitations when we are going to use the sound to determine the property of the food. So electrical properties depend mainly on the moisture content of the nature of the water held. Chemically bound water exerts less influence than free water, in which polar molecules can orient freely with the applied electric field. So moisture content of the food can be determined by the presence of electrical properties. We all know that any substance that is acting as an electrolyte means it is having some molecules, ions. So ions, if it is there, it will be having the ability to conduct electricity. So in the same way, the moisture content of the water which is present in your food will help to determine the nature of it when the electric field is applied. So the limitation of this method is loss of viability, and electrode should touch the sample. The electrode which we are using, electrode is nothing but the one in which we will see in the pH meter. pH meter, you will have an electrode. In the same way, you will also have the electrode to determine the electrical property of a component. So these are some of the limitations in the electrical property. And coming on to the radiological property, electromagnetic spectrum from X-ray to infrared have been used for various applications. So these using the electromagnetic spectrum or from the infrared using IR, they will be determining the quality of some of the foods, such as egg sorting. Egg sorting, they will do the good egg and spoiled egg sorting is done with the help of this radiological property. And apple defect detection, real-time inspection of citrus, checking firmness of fruits and vegetables, quality measurement of cheese, pork, and meat grading. All these are being done using the radiological property, that is with the help of your infrared radiations. So instrument, apart from these physical and the subjective and the objective analysis, we also have some instruments that is used to detect the quality of food. One is your gas chromatography or flometry, and it is based on the sensory evaluation and identification of aroma active compound is possible. So it is used to identify the aromatic compounds which is present in the food. Next one is your spectrophotometer. In the spectrophotometer, we will measure the concentration of solutes in the solution by measuring the amount of light that is being observed by the food. So it is depending upon the wavelength and the peak which the depending upon the formation or depending upon the presence of product, you will get the peak, and from the peak you can determine this compound may or may not be present. Next is your electronic noise. Selective for certain volatile compounds, the system is capable of identification of simple and complex odors. Odors is a smell, so it is used to determine the smell, and it is used for mainly the detection of aromatic compounds in the food industry. So if you take this instrumental techniques, it is mainly used in the food industry where the direct manufacture of food is taking place. Coming on to the most important topic apart from your food quality testing is your food adulteration and analysis. So we all know that the food which we are consuming today is not completely a pure one. It is adulterated with some or less amount of the compound. So here in this topic we'll be seeing what are the common adulterants which is present in the food and what are the ways to detect some of the adulterants. So adulteration is nothing but addition or removal of any substance from your food. Not only addition, if you're going to remove any substance from the food, it is also called as adulteration. So if you're going to add anything to the food, it is also called as adulteration. So what will happen when you are adding or removing any compound or any substance from the food? The natural composition and the originality of the food is being affected. So when you are spoiling the naturality of the food, it is called as adulteration. So food adulteration includes mixing, substitution, concealing the quality, putting up of decomposed food for sale, misbranding or giving false labels, and addition of toxicants. So all these are coming called as adulteration. So in adulteration, we will be mixing a similar compound to the food, or when we are adding any coloring agent to the food. So when you are going to substitute in the sense, instead of this you are going to add that one, that is called as substitution. That is also spoiling the quality of the food. So when you are using decomposed food for sale, it is kind of adulteration. Recently also there was one news which was going on in a shop in Chennai. A person who is using for juices, fresh juice, no, he is selling the fresh juice. So he obtained all the vegetables and fruits from the market, from Chennai only, from the main market in Chennai. So the fruits and vegetables he obtained was extremely spoiled, which was eaten by rats, and he was using that kind of fruits and vegetables to prepare fresh juices for people consuming it. So the quality inspector checked the food and he finally sealed that one. This was a news which I recently saw. So these are selling of decomposed food, misbranding or giving false labels. So if you take false label in the sense everything will be similar. For example, if you take a brand called as Pepsi or Coca-Cola or Mazda or whatever it is, if you take the label of that particular compound or that particular bottle, and if you are going to find out the false labels, means everything will be same. The color, the design, everything in the label will be same. But when you notice it keenly, we can see the difference in the spelling. There will be the spelling difference, one letter addition. So all these are called as false labels. So an addition of toxicants, instead of using the normal food coloring agent, if you see peas, nowadays we are getting green peas. If you see in a small packet, which is coming in a small packet, this is highly added to the green color. If you are taking and washing it, you can see the green dye coming out of it. So when you are consuming all these kinds of foods, ultimately some of the organ damage or organ failure may occur. So a food is declared to be adulterate if a substance is added which depreciates or injuriously affects it. Second, if it is cheaper or inferior substances are substituted wholly or in part. For example, you are going to manufacture, for example, you are getting a food or getting a product from the market. You can see the originality. So whatever food items are being exported from our country to outside India, it may be of high grade quality. Even we will say, in our country we are not getting this kind of compound or this kind of food, but whereas in the other country we are getting that one. So what they will do, or they might do, is that they will remove some of the components from the food and they will sell it to us. So the compound which we are going to add to it is of very less cost, so that they can run the business. This is one kind of adulteration. Next, any valuable or necessary constituents has been wholly or in part abstracted. So for example, if you take some so many foods or so many vegetables, what will happen? Some vegetables are rich in, all the vegetables are rich in nutrients. So if there is one particular compound which is highly in need, people, what they will do, they will extract it. They will extract it, or they will take out the necessary component from the food, or they will take half of it and they will sell the remaining. So not only in any part, anything. So when we are taking the compound from it wholly or partially, it is also called as adulteration, and it is an imitation. So we all know that we heard of news that there is a plastic rice, plastic cabbage. All these are imitation. Imitation is, it is very similar to your natural thing, but it is not natural, but it is a imitation of that one. And it is colored or otherwise treated to improve its appearance, or if it contains any added substance injurious to health. So as I already said, food colorants nowadays they are using. For whatever reasons, its quality is below the standard. If the quality of the food is not up to the mark, it is also considered as adulterated food. So if you take adulteration, there are three different types of adulteration. One is intentional adulteration, incidental adulteration, and metallic adulteration. Intentional adulteration is, it is done purposefully, intentionally, it is done intentionally, and it is done for the financial gain. An addition of sand, marble chips, stones, mud, and other filth, talcum powder, chalk powder, water, mineral oil, and harmful color. All these are done intentionally, and this is called as intentional adulteration. All these are done purposefully for money. Next is your incidental adulteration. It happens due to carelessness and in the lack of proper hygienic condition during processing, storage, transportation, and marketing. Example, pesticide residues, dropping of rodents, larvae in foods. So how these rats are biting, if it is not stored properly, it will eat the food. So all those are considered as incidental adulteration. Next is your metallic adulteration, occurs due to arsenic from pesticides, lead from water, effluent from chemical industries, tins from cans. So these are metallic adulteration, the heavy metals that has been mixed with food. So how these heavy metals are mixed with food? If you take some components like arsenic, it is from your pesticide, and lead is from your water, effluent from chemical industries, and tins from cans. Some foods which are stored in cans is adulterated with your heavy metal tin. So all these are some of the methods of, or some of the kinds of adulteration. So now we will be seeing some common adulterants and the diseases caused by them. Cow or buffalo milk can be adulterated with starch, milk powder, urea, formalin, and detergent. So what is the health defect it will cause? It will cause you cancer or acute renal failure. And how can we detect the adulteration? For example, you can take a small amount of milk, and if you're going to add if you're going to shake it, or if you're going to shake it thoroughly, you can see the formation of foam. If it is foam formed, we can say that it is adulterated with your detergent. And pure milk will form only thin layer, whereas this is your adulterated milk. You can see the difference in the picture. And here, if the milk is adulterated with starch, and when you're adding iodine, the color changes to blue. So if it is not adulterated with iodine, there will be no color change. Even now, what they are doing, they are overcoming all these adulterant methods. So we will not be able to find out this adulteration that has been caused. So they are using so much of chemicals so that to prevent, even when we are doing all these tests, we will not be able to find it. Next is your ghee. Ghee essence is used in cheaper oils and passed off as pure ghee. And this type of ghee will not solidify like the normal ghee. So if you take ghee, once it is melted, it will be solidified. And if it is not solidified, it is called as adulterated ghee. So it may also not have grainy texture of pure ghee. And if you see, the common adulterant is oleomargarine or lard, which is called as animal fat, that is added to the butter. Sometimes mashed potatoes, sweet potatoes, and other starches also added to ghee. So when you are having any potatoes, starch, or put anything, your iodine is the only component that is used to test. The color of it will change from normal to blue. If it is changing to blue, you can say that it is adulterated. If it is not, you can say that it is a pure one. So the health effect is cancer and again acute renal failure. Sometimes they will use an oil called as argemone oil. It is also adulterated to ghee, but it is highly toxic and it causes a disease known as dropsy, fluid collection in some parts of the body, and it may also paralyze the limbs. Dropsy means your water accumulation in the body is called as dropsy, and it may also paralyze the limbs. So this is the argemone seeds which I was telling. So it is also been contaminated or adulterated with your mustard seeds. So adulterant argemone or argemone seeds or argemone oil. Mineral oil also added to edible oil. So for mustard, so this is the argemone plant which we so many of us would have seen. So in the what in the dry areas you can this plant, and from this you can find out the 1 second, please. I'm sorry. Sorry for the disturbance. Okay. So this is the argemone seed which is being contaminated with your mustard seeds. And if you the method to find out this, we are going to take a small quantity of mustard seeds in a glass plate, examine visually from the argemone seeds, so you can see the difference. So mustard seeds will be plain, the surface of it is will be plain, and if you take the argemone seeds, it will be a rough texture. So from this we can see the difference. And when we are pressing the mustard seed, it will be giving you the yellow color. If you are taking the mustard seed and if you press it in hand, it will give you the yellow color, whereas if you press the argemone seed, it will be giving you the black color, but white inside, it will be white in color. So next is your sugar and salt. So adulterants in your sugar and salts, which we all know, is a chalk powder, white sand, washing soda, plastic crystals, urea, white or yellow color, rava, suji, stones, etc. So chalk powder is not toxic, but when we are consuming it for a longer period of time, it will give us a respiratory problems. And washing soda causes diarrhea, nausea, and vomiting. So this is a detecting the adulterants in your salt and in your sugar. If you're going to take the salt, iodized salt, when you're mixing it with water, if it is not adulterated with any of these compounds, it will. But if it has been adulterated with any of the compounds like chalk or some other thing, the same goes with the sugar also. Sugar, if it is not adulterated, you are going to take a sugar sample, and if it is not adulterated, it will be like this. If it is adulterated, you can see some of the components which is being settled down at the bottom of the glass. So from this we can find out whether it is adulterated or non-adulterated. Next is your tea powder. Tea powder is the most common adulterated is your dye or artificial color or iron fillings. Sometimes the fillings of ions. Sometimes what they will do, they will take the used tea leaves, they will dry it, and again they will mix it with the fresh tea leaves to increase the weight. So what will happen? Tea leaves are often adulterated with iron flakes to increase their weight. The chances of adulteration are more if we buy loose tea. So loose tea, instead of that branded packed one, when we are going to buy all this kind of loose tea, the adulteration, this iron used tea from tea stalls is dried in the sun, mixed with coal tar dyes and some amount of genuine tea to give it a flavor. So health effects, again it will cause you the tetanus and cancer. So what is a method to test it? If you're going to take a tea leaf in the paper, and if you're going to sprinkle some water on top of it, and if you're going to press it with the paper, you can see the difference. If it is pure, there will be no color emission or nothing will be around the paper. If it is been adulterated, or if it is any adulterated with coal tar, you can see the essence which is coming out of the tea. From this you can find out whether it is adulterated or non-adulterated. The same thing which will go with coffee also. So it is mixed with chicory powder, ground tamarind and date seeds. So in this we can say this chicory powder is not an adulterant, but sometimes people intentionally so much amount of chicory is also injurious to health. So apart from chicory, you can see the tamarind and date seeds. It is ground into a powder and it is mixed with your coffee powder that ultimately causes diarrhea. And coffee is also mixed with cheap ingredients like maize, soybean, sugar and acai seed. Aren't harmful, but they can spoil the quality of the coffee. Next, chili and turmeric powder. Sudan red chili. In chili, we will be seeing the Sudan red color. Sometimes Kashmiri chili powder will be very red in color. Sometimes normal chili powder is very red in color. You can see because of the presence of Sudan red, it is an artificial coloring agent. Red brick powder, grit, sand, dirt, and non-permitted colors and sawdust. All these are being mixed with your turmeric and chili powder. So, what are the health effects? Sudan red dye is your carcinogenic agent, and brick and sawdust is your causes stomach disorder, and it is adulterated with yellow aniline dyes, non-permitted colorants like methanol yellow. Yellow is for turmeric powder and lead chromate powder. So health effects, again the artificially used dye are carcinogenic, and tapioca starch, tapioca na maravalli kazhu. So they will use the powder of the tapioca starch to that will cause the stomach disorder. And lead chromate is added to turmeric powder that causes anemia, paralysis, brain damage, and abortions. So this is a method to check the contamination. If it is a pure chili powder, even when you're dissolving it in water, it will not form or it will not give you any other color. It will just go and settle. If it is contaminated, you can see the sawdust and added color that has been spreading into the water. Likewise, in the turmeric powder, if it is natural turmeric powder, it will give you light yellow color settling and the powder will be settled at the bottom. If it is adulterated, it will give you the strong yellow color while settling down at the bottom. So coming on to poultry and fish, you can see the common thing which is used to protect all these meat and fishes are your formalin. Formalin is a type of chemical that is used to keep the food fresh for a longer period of time. But formalin causes cancer, asthma, and other skin diseases. Unhygienic meat and meat products can cause food infection, usually with mercury contaminated fish can cause brain disease. Most commonly used chemical as an adulterant to protect or to keep the meat fresh for a longer period of time. Coming on to the testing procedures, microbial testing procedures, we can also see that we know that there was one more case where the batter, the normal dosa and idli batter, which you are getting it from outside, contains higher amount of E. coli. That was one research. They showed that all these batters, which we are procuring it from the shops from particular area in Chennai, has larger amount of E. coli. E. coli is also pathogenic when it is in the when we are exposed to a larger amount. So all these kinds of batter or microbial testing procedures can be done by the help of these methods. So examination of food for the presence of microbes and their metabolites are called as microbial testing procedures. In some case of pathogenic microorganisms or their toxins, you simply want to know whether they are present or not. So just to detect whether this organism is present or not, we can use the microbial sampling procedure. First is a surface sampling. It is a swabbing. Swabbing, we all know. We will just take the petri plate, prepare with the agar, and then we will swab the food product. We will just take dip the swab in the food product and we will just rub it on the plate. Next is your contact plate, raised agar plate that is pressed against the surface and then incubated. We are just going to prepare a plate. We are just going to expose the plates to the top of the food surface and then we will incubate it. Next is your excision method. A plug of known surface area is taken from the food and it is directly incubated in the plate. Okay, so homogenized and plated into to determine the total number of colonies, colony formation. So for example, commonly used is whole cut meat. So we'll take a small piece of meat and just we will plate it in the we'll place it in the plate, petri plate, and we will observe for the growth of microorganisms. This is called as surface sampling. Next is your homogenization methods. Homogenization means we are going to break or we are going to grind the following foods into small pieces. That is your blender. We will blend it and then we will go for plating. Next, Caldwell stomacher. Food is placed in a sterile plastic bag with dilutants and then inserted into the machine. The stomacher has two needles that vigorously disrupt the food and give a nice homogeneous fluid for sampling. That fluid alone we will take and we will go for culturing. So next is your genetically modified foods. So genetically modified foods are produced from organisms that have changes introduced in their DNA by using the method of genetic engineering. So we all know there are so many genetically engineering foods which we are using. Golden rice, tomato, alfalfa, sweet corn, summer squash, canola oil, salmon fish is one of the genetically modified food. But some of these genetically BT brinjal, BT cotton, all these are some of the genetically modified things. Some of them have been approved by FDA and some of them are been waiting for FDA approval. So genetically modified foods in India is all these: golden rice, BT brinjal, BT cotton, small cherry tomatoes. This is called as cherry tomatoes. This one is called as cherry tomatoes, and this is a banana which we consume, which we can see in market we will be consuming day to day, but it is highly not advisable to consume this kind of banana. Coming on to BT brinjal and BT cotton, we know that there is a bacterium called as Bacillus thuringiensis that is used to reduce the pest that is affecting these kinds of plants. So these are some of the genetically modified foods that is used in India. Coming on to the food safety certification, we all know that we have seen what is meant by food testing, what is meant by food quality, and what are the kinds of adulterants which we have seen. And now we are going to see the certification. What are the agents or who are the people who can certify the food which we are consuming? Food standards, that is called as food standard. Food standards are something that are set up by experts or an authority for measuring quality, weight, and value of the food. So international food standards, we have Codex Alimentaris Commission. It is a principal organization of the worldwide food standards. So apart from FAO and WHO, we have Codex Alimentaris. And apart from that one, we have two more agents called as Food and Agriculture Organization. Next is your World Health Organization. And if you take the food standards in India, it is coming under Codex Alimentaris. There are two standards. One is compulsory standard and the other one is your voluntary standard. Compulsory standard is whatever food which we are going to consume, it should have this kind of standard. Voluntary is apart from compulsory. If you want your product to be tested for this thing, it depends upon you. So compulsory standards: Prevention of Food Adulteration Act, Essential Commodities Act, Fruit Products Order, Meat Products, Milk and Milk Products, Solvent Extracted Oils, Flour Control, Vegetable Products, Standard on Weights and Measures. All these are called as compulsory standards. So all these are mandatory for any kind of food. All the food products have to undergo any what are depending upon the product, it has to undergo all these kinds of compulsory standards. Voluntary standards is nothing but your AGMARC, BIS, ISI. AGMARC we all know, Agricultural Producing, Grading and Marketing Act. It is the abbreviation of your AGMARC. So direct marketing and inspection grades of commodities in 1234, meaning special food, special good, fair. It is 1, 2, 3, 4. Depending upon the one means it is special, 2 good, 3 fair, and 4 ordinary. The AGMARC label has been the assurance of quality. So it also helps us to satisfy the buyers and sellers. Next is Bureau of Indian Standards. Indian Standards Institute is responsible for laying BIS. So BIS, if you want to get BIS mark, it is a institute. ISI is the one which is giving you the BIS mark. These standards are evolved after chemical, biological, and physical assessment physical, chemical, and biological property, and it will give you the standard. Next, challenges in food safety. So there are some of the challenges in food safety: changes in our food production and supply, including more imported foods, changes in the environment leading to food contamination. So what are the challenges? Why we can say that there are some of why our food has to undergo all these kinds of quality testing? Because there are so many changes that is occurring in the production methods, supply methods, and import of foods. So to overcome that one, we have to undergo the food quality testing and food standard. Changes in the environment leading to food contamination, better detection of multi-state outbreaks, new and emerging bacteria, toxins, and antibiotic resistance. So whatever it is, when we are going for microbial testing, we can see so many new species of microbes which is evolving. It is releasing so many toxins, and some microbes are really antibiotic resistant. Whatever antibiotic we are consuming, it is not going to die, it is becoming resistant. So to overcome all these and to reduce the effect of all these things, we have to undergo the food safety and food standards. So changes in consumer preferences and habitat. It is not that every day a same kind of food or same kind of people will be consuming it. It differs among people to people. So to overcome that also, we should, all these are some of the challenges in the sense why we have to do the food quality and food testing, why it has to be done to satisfy the people first, and to prevent ourselves from some of the foodborne illness. And for all these things, these are the methods that has to be followed, that is food standards, food safety, food quality testing, everything has to be followed. So apart from this, we can have now this shows you where you can have a study about food quality, food testing courses. These are some of the institutes in India which is providing you the food testing courses. The Cashew Export Promotion Council of India. This is just for your idea. Some of you wanted to go for food quality inspector, all this. If you can go and browse these websites, they can tell you on how to apply and what is the course duration, everything will be given there. So Institute of Good Manufacturing Practices India, Food Safety and Standards Authority of India, Indian Institute of Crop Processing Technology, and Bureau of Indian Standards. So this is some of the food testing courses in India. And with this, I would like to thank the management for giving me an opportunity to present this talk. And I would like to thank Dr. V. Kokila for organizing this one.
Thank you, man, for your interactive session. You have thrown a light of the topic.
Thank you.
Now it's time for questionnaire section. If anybody having questions, kindly raise your question in the chat box. Thank you all. I would now like to invite Miss R. Deepika from Department of Plant Biology and Plant Biotechnology to deliver vote of thanks.
Greetings to everyone present today, including our distinguished chief guest, Dr. J. Vardhana, and esteemed principal, Dr. N. Sujata ma'am, and all the participants. It is an honor to have the opportunity to express my appreciation to all the dignitaries present. I am grateful to the esteemed delegates of the webinar for their presence and efforts in making this event a success. I extend my sincere thanks to our honored chief guest, Dr. J. Vardhana, for taking time from her busy schedule to be with us. A special thanks goes to our esteemed institution management and our respected principal, Dr. N. Sujata ma'am, for their unwavering support in making this webinar a success. My gratitude also go to Dr. B. Kokila ma'am, coordinator of Enviro Club, for her efforts in organizing this event. I also thank Dr. C.P. Prakash sir and Dr. B. Bharat sir, assistant professors, Department of Plant Biology and Plant Biotechnology, for their constant support. I must also acknowledge our public relation officer, Mr. Balaji sir, and esteemed members for their hard work in the lead up this webinar. I extend my thanks to all the active participants in this event. Thank you once again to everyone for making this event a great success. Thank you.
Thank you, Deepika. Once again, thank you for all the active participants and dignitaries. We will end the session now.
Thank you. Thank you so much all.
Thank you, Vardhana.